Good evening, everybody. I hope you guys are all doing well. Welcome to week 7, Bio 1022, the nervous system. Now, as with all of these, I'm going to try and go straight through, but this is a long one. There's going to be, so please pause the video, pause the PowerPoint, watch the videos, go back, read the textbook. This is a good one, and this will take you a while, so pull up a chair and get a coffee. We're going to be a while. So let's get this going, shall we? The nervous system, basically here we're talking about communication between your brain and your big toe, and there's two ways to do that. There is the nervous system, which is fast, efficient, but short-lived. There's also the endocrine system. Here we're talking hormones, and this can be a slower way for the brain to talk to the big toe, but the effects can be monumental and a lot stronger and a lot longer than the nervous system. So two different ways there, but today's lecture is on the nervous system, which is made up of the brain, spinal cord, and these nerves that come off the spinal cord there. Okay, so if you have a look down here, you have the central nervous system, which is the brain and the spinal cord only. You have the peripheral nervous system, which is the nerves coming off. Okay, and the whole idea there of sensation coming, now let's say you burn your hand, the sensation comes up, okay, and travels through the peripheral nervous system, these nerves, to the central nervous system where a decision is made. Okay, whether it goes to the brain or stays in the spinal cord, and then an effector nerve comes down and produces a response. Okay, so you look here, the nervous system is organized to detect change in internal and external environments, evaluate the information, and initiate an appropriate response to that information. Detecting sensory stimuli, analyzing and integrating sensory information, responding to internal and external stimuli, regulating organ systems, homeostasis. Most sure the body's perfect, the outside environment is not, so the internal body has to maintain its particularities, and this is done through regulation and homeostasis. Reflexes, rapid, unconscious responses to stimuli, learning, memory, and emotions. All right. As we said there, the central nervous system is composed of the brain and the spinal cord. The peripheral nervous system is composed of the nerves that come off of the spinal cord there and reach out to all different parts of the body, right? The basic building blocks of the nervous system is the neuron. That's the basic cell. And so the central nervous system, structural and functional center of the entire nervous system, consists of the brain and the spinal cord. The peripheral nervous system, nerves that lie in the outer regions of the nervous system. Cranial nerves, spinal nerves, they originate from the spine. You have the afferent, these two are good, afferent and efferent. Any nerve that is incoming, at home, afferent, incoming from the outside into the central nervous system. Efferent nerve is an exiting nerve. It's coming from the central nervous system away. It's an exiting nerve, efferent nerve. And this is what we'll be talking about there today, the overview of the nervous system, okay, which is broken up into the CNS as opposed to the PNS, okay. Central nervous system, brain and spinal cord, but the peripheral nervous system, now we're into what's called the somatic, which is body, okay. We have control over our skeletal muscles. That's what's going on here. But these two here, I don't have any control over. The autonomic nervous system, this one here, is broken into what we call the parasympathetic, rest and digest. You can have a sleep, you're okay, your life is not in danger. You can digest a banana, you can go to the bathroom. If your life is in danger, you are now in fight or flight mode. Okay, if a Tyrannosaurus Rex is in the bedroom and it's going to eat you, well, you don't have to go pee. You don't have to, that banana that's in your stomach right now stops digesting because your life is in danger. We'll talk about these ones here. And there's also the enteric. This one here has to do with the organs themselves. So the somatic nervous system, voluntary control of movements. Afferent neurons at home bring signals from the outside to the inside. Peripheral nervous system to the central nervous system. Efferent neurons bring signals from the inside central nervous system to the outside skeletal muscle fibers for a resultant action. Autonomic nervous system, involuntary control of smooth and cardiac muscle and glands. We don't have control over our stomach or peristalsis. Once we swallow that banana, we don't hear hide nor hair of it for another 30 hours or so. This system works with the endocrine system to maintain homeostasis. The enteric nervous system, it's also part of the autonomic nervous system. These are neurons that regulate digestion. Submucosal plexus stimulates secretions there. The gastrointestinal system. Myenteric plexus stimulates smooth muscle contraction. The whole idea, there's your afferent and efferent. Now, just going into a bit of detail here, you see that if there's some type of stimuli, you burn your hand, that information travels through the afferent neuron at home coming towards the central nervous system, and then it hits an interneuron. Now we're into the central nervous system for sure, where a decision is made, and then the resultant message is traveled along an efferent neuron where an effector, the result, you move your hand away. Okay. Here, the somatic nervous system, if you smell a cheeseburger from McDonald's, okay, you're driving by, and you know how they always have the window open, especially Kentucky Fried Chicken, right? You can smell that for miles. So some type of sensory receptor receives a message that travels down, okay, an afferent neuron, okay, to an interneuron. Here we're into the central nervous system, and a resultant is given. Is sent down the efferent neuron, the effector skeletal muscle, and now I am running to Kentucky Fried Chicken. Okay. When we're talking the autonomic nervous system, it's kind of the same thing. It takes a different pathway, but the resultant is the same. There's a sensory receptor, interneuron, and in this case here, the effector organ, smooth muscle, cardiac, whatever, your heart, whatever going on there, okay? Good. This one here is a great slide. I took up this is how the nervous system works in a nutshell. And as long as you start here, so this is your sensory division, and the whole idea here, where it's going to be kind of the same way. There's some type of your sensory neurons there. They detect stimuli. So the sensory afferent division, here we're talking somatic and visceral sensory nerve fibers. They conduct an impulse from the receptor through the peripheral nervous system, right? Because if you detect something, that message has to travel through the peripheral nervous system to the central nervous system, right? So that's what's going on here. It travels through the peripheral nervous system, that stimuli, to the central nervous system where a decision is made of some kind. And then now we need to see the effect so that the interneuron the message gets sent back onto an efferent. There's your motor division through the peripheral nervous system to an efferent nerve. Okay, exit. It's now leaving. It's going away from the body. And if that elicits a response that we can have control over, then that will go to the somatic nervous system. Okay, where I'm running to Kentucky Fried Chicken. But if there's a dinosaur in the bedroom and it's going to eat me, well, I sense that. Message goes through the peripheral to the central, down to the motor, but it goes here now. If my life isn't threatened, I don't have control over what's going to happen. All of a sudden now my body kicks into what's called the autonomic nervous system. Now, if that dinosaur is going to eat me, then you guys have all heard of you hear about this every once in a while, where like a 50-kilogram mother sees that her child is trapped underneath a car. Well, then that mother will end up lifting up the back end of a Honda Civic and saving her child. Well, we are all superhuman. We all have that, and that's called the sympathetic nervous division there. Fight or flight. If our life is in danger, we can become superhuman. Okay, along with the autonomic nervous system branches into the fight or flight, or it could also branch into what's called the parasympathetic. This is rest and digest. Okay, when our life is not threatened, we're able to go to the bathroom and digest a banana. But we'll get back into this there later on. But this is the central nervous system. This is the nervous system for the body in a nutshell and how it works. It receives sensory information, and what it does with it can go a number of different pathways there. Overview of the nervous system. Neuron, a single cell. Okay, nerves there, they contain thousands of neurons that connect with a specific tissue. Glial cells that support the neuron with the central nervous system and PNS. Neurons do not divide, but we do have some stem cells that we can differentiate into neurons if needed, but generally, if you damage nerve, it doesn't come back. If you break your back, you can become paraplegic or quadriplegic or some type of nerve damage. You will hear a plexus ganglion sensory receptors there. Plexus is the network of neurons and nerves that innervate a specific tissue, such as submucosal plexus there for the digestive system. Ganglion there, a group of cell bodies, not the axon or terminal, but we'll get into these in a couple slides there in the nerves, such as the dorsal root ganglia that brings sensory information to the spinal cord. Sensory receptors detect specific sensory stimuli. Can be special cells that connect to a sensory neuron, can be extensions of a sensory neuron. And you'll see there's different things there that can be sensed. Okay, and these receptors here are on all the time, and they're monitoring, right? We have chemoreceptors, chemical concentrations such as hormones, neurotransmitters, and nutrients. These are all being monitored. Osmoreceptors, concentration of water and ions, osmolarity. Baroreceptors, blood pressure monitors found in the blood, especially in the aorta. Photoreceptors, retina. Mechanoreceptors, stretch and physical, think bone, osteocytes. Proprioreceptors, body positioning, how do you know to stand up straight? Nociceptors, pain. Thermoreceptors, temperature. Tactile receptors, touch and pressure. Now, put your fingers on the table there and you can feel the table. I'm feeling it right now, but I don't have any nerves in my epidermis. The nerves are found in the dermis. So what is it exactly you're feeling? Is it the table itself, or is it just the pressure of the table pushing on your epidermis? Cell bodies. This is what we call your cell body there. Houses the nucleus and the ribosomes. Ribosomes, what do these guys make? Proteins. Interesting. Think of axonal transport. We'll get there. What proteins are being made within a neuron? Dendrites. These are these tentacles here. And if you think of this is the start of a neuron and this is the end, well, there will be another neuron over here. Okay? And you've got your dendrites, which are receiving a message from a presynaptic neuron. Okay, and if this is a presynaptic, then this would be a postsynaptic. Okay, because one neuron comes into contact with another neuron, doesn't touch it, but it creates a synapse, a little gap in between. So there would be another neuron over here. The dendrites of another neuron would be over here, and you see the message comes down, travels down the axon to the axon terminals where they have these bulbs here. Okay, teledendria, and the electrical impulse travels across to the next neuron. Okay, so you have dendrites. These are receiving the electrical signal from other neurons. The axon is down here. This is the tail that the message will travel along. But let's not forget about the axon hillock. Okay, this is where the electrical signal will kind of converge. So maybe we get a stimulus coming from here, another stimulus coming from here, and another stimulus coming from here. They will all meet here and then converge, okay, through a process called summation, which we'll get to there in a little bit. So the axon and the collaterals here, they transmit electrical activity starting from the axon hillock and moving towards the axon terminal. These collaterals increase the number of cells influenced by one neuron, increase thinking, more dendrites, and more axon collaterals, more signal being transmitted. The axon terminals here, axon bulbs, they release neurotransmitters which connect to other neurons or muscle. So there would be another neuron right here, okay, but they do not touch. So these axon terminals, they look like they touch, but they do not. And the message gets sent from one nerve to another. When the message comes down, neurotransmitters released, which then induces the signal in the next neuron. Okay, these guys here, these are what we call Schwann cells. They are myelated cells. Okay, we'll get there. There are three kinds of neurons there I want you guys to have a look at. There is the most common, which is what we call multipolar neurons. They send information to skeletal muscle or smooth or cardiac. Okay, they're efferent, moving away from the central nervous system, motor neurons, interneurons. Okay. Then we have unipolar sensory neurons here. They send information from the receptor to the CNS. So these guys here are afferent. Okay. There are interneurons, very short connecting neurons in the central nervous system. We've seen those already. Okay. Tend to be multipolar. And then there is bipolar. And you'll see the difference there between unipolar. They kind of look the same, but the unipolar, the cell body is sticking out. When you're talking bipolar there, the cell body is connected straight on. It's in the middle of an axon. Okay. And these are found in the ear, retina, olfactory areas of the brain. The neuron is exactly two extensions. Okay. Sensory, the least common. Okay. Now we're into glial cells of the central nervous system. You can see there's astrocytes here, microglia. These ones here are phagocytic, ependymal cells, and oligodendrocytes. So the astrocytes here regulate extracellular fluid, ions, and neurotransmitters, guide neuron growth during embryonic development, involved in formation of the blood-brain barrier. That's right, the brain never touches blood. There's another organ as well, the testes there. There's also, it never touches blood. It's just a way of safeguarding the brain. While the blood is great, it brings oxygen, nutrients, all these kinds of things. Blood can also bring bacteria. So it's just another way of removing the brain from being contaminated, okay, potentially, right? Stores glycogen, forms scar tissue. Microglia, these are phagocytic immune cells, can migrate to infected areas and engulf pathogens in dead cells. Ependymal cells they're often ciliated, involved with production of cerebral spinal fluid. Okay? And oligodendrocytes make up myelin, very high fat content, protect and insulate the axons for sure. Okay? Glioma, these are brain tumors, are usually formed from glial cells. Talking about these glial cells when you're talking the peripheral nervous system, okay, if you go back and you'll see there these myelated, these area of the axon that's covered there by these what we call Schwann cells. These are myelated, okay, and they're involved in what we call saltatory conduction, which we'll get into. The satellite cells surround the neural body located in the PNS, similar to astrocytes, and function of regulation of extracellular fluid ions. Okay, Schwann cells here we go, surround and form these myelin sheaths around the larger nerve fibers, similar there to oligodendrocytes. And as well, in between the Schwann cells, we have what's called these nodes of Ranvier, these gaps in between the myelin sheaths. Okay? And when you're talking saltatory conduction, the nerve impulse actually travels faster with the Schwann cells in place, because the signal will, instead of traveling all along here, the signal goes jump, 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 jump. That's what we call saltatory conduction, and the signal travels faster, okay, because of these myelin sheaths. And you see how they're wrapped around here, okay, and the gaps in between these are called the nodes of Ranvier, these gaps in between the myelation. Parts of the neuron here, you'll see here that this is the cell body, also known as the perikaryon, okay, ribosomes, rough endoplasmic reticulum, okay, Golgi apparatus for packaging. Why would we need to package things up? Neurotransmitters are packaged into vesicles. They provide protein for maintaining and regenerating nerve fibers. Interesting. So neurotransmitters are made, and what? They're sent down because that's where they're going to be released at the end of the axon. These are dendrites. These are receiving messages from presynaptic neurons up here, right? Each neuron has one or more dendrites which branch from the cell body. The distal end of the dendrites of the sensory neurons are receptors, okay? Now, just continuing on here, it's the same, continued there, next slide. You'll see there that the axon, just continuing on here, conducts the nerve pulses away from the cell body of the neuron. So the nerve, the impulse is traveling along, and eventually it meets the end. And you'll see here that there are these little arms at the end called telodendria. And then at the end of these there you have these axon bulbs, okay? A single process extending from the axon hillock, sometimes covered by a fatty layer of myelin sheath. These are Schwann cells, okay? Axon varicosity, swelling that makes the contact synapse there with other cells. The distal tip of the axon forms branches called telodendria, each of which terminates in a synaptic knob. Whether an axon is myelated or not also affects the speed of the impulse, saltatory conduction. And you'll see here that when we're talking about saltatory conduction, it refers to the action potential that occurs in the neurons that have myelin sheaths covering them. Only the nodes of Ranvier have the ion channel that opens and allows ions to move in or out of the cell, and it dramatically increases the speed of conduction, okay? And you'll see here that this is all controlled by these ion channels. Only the nodes of Ranvier have these ion channels, because the whole idea of electrical conduction is when you're talking the neuron, you're going to have basically a little battery. And it costs us money. That's why we have to eat food, is that because in order for these neurons to work, it has to be positive on one side of a membrane and slightly negative on the other side of a membrane. And once we have a positive and a negative separated by a membrane, you basically have a little battery, potential energy, you're able to do work. And that's where we're talking about here. These ion channels, sodium-potassium pump, possibly you've heard of that. Here we're talking about the nerve itself. There's a couple of coverings there that I would like you guys to know. You see on the outside of a neuron there, we have the epineurium. You have the superficial as opposed to the deep epineurium, okay? And then if we, just like in muscle, you remember there, these fascicles, they pop one of these out, okay? And what do we have? We have the perineurium, which is the outer covering of one of these guys here. And then just on the inside of there, you have the endoneurium. And if you uncover that, then that's where the Schwann cells, that myelated sheaths, are. And then you have all of your axons kind of in a row. They're all parallel. So each nerve contains axon bundled into fascicles. That's what these guys are. A fibrous endoneurium surrounds each axon and its Schwann cells within a fascicle. The perineurium, okay, surrounds each fascicle, forming a blood-nerve barrier. A dense connective tissue epineurium wraps the entire nerve. Okay, and you'll see here that you see how these myelin sheaths are wrapped around. That's exactly what's happening. Next slide will show you it even greater. Okay, and you see the myelation. These are the Schwann cells, okay, and they're literally wrapped around. And if you zoom in on this electron micrograph, you will see the exact same thing of what's going on. You know, that myelation sheath is wrapped around and protecting the neuron, the axon there. The diagram shows a cross-section of an axon and its coverings formed by a Schwann cell, okay, and this is an electron micrograph of a real one there showing you it in detail, okay. This one here I really like. The whole idea there kind of connects the whole neuron in a nutshell with this little cartoon caricature. You'll notice there that there are ribosomes that we've talked about, the Golgi apparatus. We're making protein up here in the cell body and we're packaging that, whatever it is. Well, whatever it is called a neurotransmitter, and we're packaging this protein and delivering it down to the end because we don't use the neurotransmitter at this end, the dendritic end, we use it at the axonal end. Okay, right? These microtubules, microfilaments, and neurofibrils, bundles of neurofilaments, allow the rapid transport of small organelles. Small motor molecules attach to mitochondria and vesicles containing neurotransmitters and carry them all the way down to the end. The used vesicles and transmitters are then returned to the cell body by the same process but in reverse. This is called axonal transport. So you'll see synthesis and assembly and export from the cell body. You're making these proteins. There's your Golgi apparatus that we're going to use to package these proteins, these neurotransmitters, and then they are sent, packaged, and sent down where they are released. The release of the neurotransmitter, which is used, okay, and those neurotransmitters, they go and they induce the conduction in the postsynaptic neuron, but then all of those neurotransmitters are reuptake and used again, where they are recycled and sent back up, where they are repackaged and sent down again. Okay, axonal transport. The action potential and graded potential. This is a great little movie on the whole idea of how a nerve is conducted from one neuron to another and all the way down the axon. And it's you need to have ions. And I don't know if you heard about the actress there. I think her name was Brooke Shields. She was on set for some movie and she was drinking water, and she almost died because she diluted herself so badly that her ion concentration was so low. You gotta have salt. I know salt gets a bad rap, but you gotta have it. The ion concentration in the neuron cytoplasm and in the interstitial fluid are not the same at all. There are more sodium and chloride ions in the interstitial fluid, and there are more potassium ions in the cytoplasm. The charge inside the membrane of a cell is negative relative to the outside of the cell. So the outside is positive, the inside is negative. Now, it's not a normal situation, right? If things were left to diffusion, you'd have the equal number of charges on inside and outside, but it's not. We eat food to maintain this difference, and it's what we call the sodium-potassium pump. Okay, the potassium moves a little faster, and then the sodium as well, but we'll show you these there. What is the end result of an action potential? An action potential is just the conduction of the message down a neuron and to another neuron. So another neuron, which they don't touch, they're separated, but the ability there of a message to move along a neuron down its axon and move on to another neuron. The charge changes cause neurotransmitters to be released from the axon terminal. These are signaling molecules that stimulate another neuron or a muscle cell. And you'll see here that all living cells maintain a difference in their concentration of their ions across the membrane. And this leads us to what we call membrane potential. Potential energy is equal to a battery. And as long as you have, a positive charge on one side of a membrane and a negative charge on the other, you have the potential to do work. So there is a slight excess of positive charges on the outside of a membrane and a slight deficiency of positive charge, negative charge on the inside. Neurons are remarkable among cells because they initiate and conduct electrical signals called nerve impulses. A nerve impulse can be described as a wave of electrical fluctuation that travels along the plasma membrane. Now you'll remember there when we talked about muscle, the whole idea that when, like in this case here, we're not talking muscle, we're talking nerve to nerve. Well, it's kind of the same procedure there when a nerve meets a muscle. And remember there, it was called the neuromuscular junction. The message from the nerve travels all the way down the nerve, down its axon, down its teledendria, down to its axon bulb, and then through the use of calcium, that message is able to get across because the calcium initiates the release of neurotransmitter, which then floods the gap in between the nerve and the muscle. In this case here, nerve and nerve, and the message transgresses and moves across the gap towards the neuron or the muscle. And you'll have a look here. If this is like a typical cell, this is the interstitial fluid, extracellular fluid there. Inside the cell there, it's all positive, negatives. In the interstitial fluid, it's all positive, negatives. But you see the membrane of that particular cell. It is positive on the outside, negative on the inside, and that's on purpose. You maintain this potential difference because now you have a battery and now you're able to do a bit of work. Okay? And how do we measure this here? The voltmeter records the magnitude of electrical difference over time, which in this case doesn't really fluctuate from minus 70 millivolts. Okay? Very slight difference in the concentration of oppositely charged ions across the plasma membrane causes a recording there of minus 70 millivolts. The voltage recorded over time is a red line. Due to the distribution of ions inside versus outside, the sodium-potassium pump will restore this and maintain this. Okay? Governed by the presence of specific membrane-gated transport channels. Okay? Allowing specific molecules to diffuse across the membrane only when the gate of each channel is open. And we'll show you these there. This has to be maintained because if this was just left to diffuse, then all of a sudden it would be plus and minus, plus and minus, plus and minus all around. But this is maintained for a reason, minus 70. The inside of all cells is approximately about minus 70 millivolts. Since the inside of the cell is negative compared to the outside of the cell, it is said to be polarized. If positively charged sodium ions move into the cell, it will become less negative. This is called depolarization. It's kind of like the same as contraction. Something's going to happen. So, this is how an action potential and graded potential work. We transmit electrical signals by moving ions across a cell membrane, changing the charge. So you see here you have sodium ions on the outside, potassium ions on the inside. In order to get an action potential, in order to send a message down a neuron, these sodiums have to come in. There's a rush. And then this potential, this recording will go up. And once we pass a certain threshold, there's no stopping it. But if we get little changes in the cell membrane, it might be a little change here, a little change there. If it didn't pass this threshold, then that will be nothing. But we have to pass this threshold in order to get an action potential, and we'll show you these right now.